All right, so the lab we're doing this week is going to be for urine analysis. So this is kind of similar to what you guys may have done in first year bio, where you tested foods for different organic macromolecules, glucose or proteins or lipids and things like that. So sort of the same chemicals will be seen. You guys will be testing for proteins. You guys will be testing for sugars, but now you're going to test for something called chlorides instead of lipids in there. We're also going to be looking at pH. So when we think about the human body, we think about what makes kidneys healthy and the purpose of urine. And we know that urine is kind of this cellular waste material that's being passed out of the body. And it will actually tell you guys a lot of information about what's going on with the body. It's one of the first things that doctors can detect. They'll actually test urine. It will show up better in urine rather than blood, is if you have different diseases, or maybe your kidneys aren't functioning as well as they should be. So it's one of the easiest things to collect. It's non-invasive in order to collect the urine. The urine that you guys are working with today is synthetic or fake urine. And what you're going to be doing is you're going to be testing a control patient, and this control patient is going by the name of Mr. Rollins. He's our control patient because what you guys are going to see in terms of his test results, they are all completely normal. So as you guys go through the particular lab, each of the scenarios that you guys have, you'll have a little patient profile. So it kind of tells you about Mr. Rollins himself, and you guys will also get a patient after this as well, and you'll get their own little profile. The patient that you'll get is going to have a problem, so you guys kind of have to detect what is going on with that particular patient based on his profile, based on his test results of that particular urine. So Mr. Rollins is age 54. He's come to the physician, just his yearly physical, to yearly checkup. Along with blood, they usually will test urine. So he's not currently complaining of any symptoms. Because of his age, urine's one of the easiest things for them to go through and collect. So a little setup that you guys have, we have these little glass tubes, we have our little chem plates that you see here, and they are on our paper underneath that is kind of blackened in. We'll talk about that for in a moment. And we have some pH stuff along the bottom. You have a data chart that you guys will be opening up. So we're going to run through control patient first. Tomorrow you guys will see your actual patient, and you guys will get a little scenario on him as well. So this should be on your Google Classroom page also. So to start out with, those who are in class, it says control patient Mr. Rollins. You guys are going to get a tube. This little gray tube is going to have a letter on it. It's important because this gray tube eventually is going to make its way to the water bath because we're testing glucose in it and Benedict solution. And Benedict solution actually needs heat to activate the solution for it to change color. So you just kind of want to write that down to make sure you know which tube is yours, because you want to make sure that you're pulling the correct tube each time. Mr. Rollins' urine is labeled, so we're just going to kind of give it a little shake, and it's going to tell you guys to kind of add about 10 drops or so into the glass tube. Now we're going to do a couple things with this glass tube. The first thing we want to do is we want to test pH, and this is something that you guys should have done in first year bio. And with pH, I put a little scale down here. We know that pH runs 0 to 14. We did it with our water. You guys will see it again with some basic chem stuff coming up. And the pH paper that we're using is pea hydrant paper. So pea hydrant paper is this orange paper. You'll dip it in. It should change kind of a color. Now, urine should be kind of, around that 7-ish. It's probably a little bit more acidic, 6-ish range, but anything from about 5 to 8 is what you guys are going to end up seeing here. You guys will take it, you'll dip it in, it should change color, and you kind of want to match it up to where we have on this particular sheet. So these go by whole numbers, so I would say, it's probably about maybe 6.5 or so. And you want to match that up pretty quickly before it dries. So I would say, okay, well, it's about 6.5 is my pH. But I'm going to go a little farther than that, because we have a second type of pea hydrant paper that goes a little bit more in detail. So this pea hydrant paper will go from 5.5 to 8. So it's giving you every 0.3 or 0.2 numbers, and it's just going to be a little bit more specific. Looks just like the other one. This one's in a roll. You'll actually just take a little piece. You're going to go through, you're going to rip it off, and we're going to kind of do the same thing. A couple drops. Hopefully we can get some on there. There we go. And now we're going to go through and kind of match that up. And we want to match that color along here. So where we said about 6.5, now we're looking at it and I go, okay, well, I'm going to say probably about 6.2. Kind of matches up with that particular color that we have there. So I'm still going to leave my 6.5. That's for my pea hydrant paper. But more specifically, he's about 6.2. I can't tell that with my normal pea hydrant paper. I can't go, yep, 6.2, because it only gives me whole numbers. I can go halves, kind of estimate it. The second type is definitely going to be a lot more accurate there. So now with this glass tube, we need to look at color. And I say kind of hold it up to something that's white. It's a little easier for you to see. And don't just go yellow. You guys will see a chart that goes through a bunch of different colors for you. And it tells you kind of what's going on. So don't just assume, oh, there's color to it, there must be something wrong. Because a lot of the foods that you guys eat will change the color of your urine. It will change the pH of your urine. It may have more sugar within your urine. So just because you see positive test results for some of these, think about the scenarios that they have or the situations that these individuals are coming to the doctors and telling the doctor, because there may be a problem and there may not be a problem based on what you have. So you'll see some colors down there. Light yellow to amber is your normal, clear to light yellow. It can be anything from alcohol that changes it, foods that you eat that have carotenes in it, like carrots, beets especially will change it, where it's going to turn kind of bright red, different foods. So you guys will see that there as well. So if we're looking at it, I would say probably, oh, it's kind of like a light urine or a light yellow in terms of that particular urine. So I would mark that under color. The next is clarity, and I want to know the turbidity of it. And what I mean by that is it clear? Can you see through it? And this you can actually see through. You'll see some samples maybe along the way that are actually very cloudy. And that turbidity or the clearness that we see within there can be caused by different things as well. And you'll see that on your data chart. So I'm going to say that it is going to actually be clear, and your results here either say clear or cloudy. It doesn't really give you too much. So I'm going to say it's clear. I'm going to say my color is light yellow, and you guys are filling this right in on your Google Classroom form. Next we have glucose, proteins, and chlorides, and it goes through your instructions on how to do this within that lab packet. I'm going to kind of go through it off my head here. So to this particular sample, this glass tube that is labeled, we are going to add 5 drops of Benedict solution. So Benedict solution, once again, is that really pretty kind of oceany blue color. And this will actually react when activated with heat and will turn either a green color if we have a little bit of sugar, or a bright orange red color if we have a ton of sugar. So you'll see on your data chart, if you have 0 sugar, it's going to stay the same color, kind of a bluish color, so we would say it would be negative. If it changes a slightly green, we'll say +1, a little bit of sugar getting passed in the urine. If it's maybe a darker green, maybe +2. If it's red or orange, it's +3, and we have a lot of sugar within that. So this would actually go off. There's a heat bath underneath the chemical hood over there. I'm going to kind of put it off to the side right now. We have one in there, so we'll come back to that. That has to sit 3 to 5 minutes. So while that's sitting in the hot water bath in there, we're then going to take Rollins urine and we're going to run and look at our proteins and chlorides. Same thing. We have 10 drops, and we're going into well number 1 here. You can actually see it right through. So 2, 3, 4, 5, 6, 7, 8, 9, 10. If we have a little more 10, that's fine. And then I'm going to add 1 to my blackened in well, and that's going to test for my chlorides. So we're testing 2 different things here, proteins and chlorides, and I'm doing them in a specific order. My sugars, that's on heat right now, takes about 5 minutes. My next one is going to be for protein. If you guys remember Urine solution from first year bio, that's what we use to test proteins. So I'm going to go through 1, 2, 3, 4, 5, and what I'm looking for is I'm looking for a kind of a chemical change there, and we want it to turn more of a pinkish or purple color. Okay, so we can kind of see it's kind of this just bluish color right now. However, we want this to sit for about 3 minutes as well. So by the time I pull that glucose tube out of the hot water, my protein should be done. I can now look at my chlorides. So I have 1 drop on there, and I put it on the black surface because if it is positive for chlorides, you're going to see a white precipitate that actually forms. It's going to kind of look really, really cloudy in there. So I'll add my 1 drop of chlorides, give it a little tap, and if that had turned cloudy, you would physically see it. And I'll put some examples on Google Classroom so you guys can see some pictures of positive test results versus negative test results. So that, I'll put it up here too, you can actually see is still clear, nothing in it. So we're going to go through. I would say my chlorides are negative there. I'm then going to go back. It should be about 5 minutes at this particular time. I would go through, I would pull my tube, and I would look at it. And I would go up, coming off the heat. Mr. Rollins, he's still the same color as he was before. There's no color change there. So what I can say about that is there's no presence of glucose. So I'm going to put a negative there. Remember, he's our control patient, so we're not expecting any within him. My proteins, we're saying 3 minutes. Nope, there's no color change there. It's still blue, and it's blue because we added urate solution, which is blue. So don't go, oh, it changed colors. It changed colors because of the solution that we added. There was no chemical change, and if there's a chemical change, it is going to turn more either pink or kind of a purple. You'll see a definite change within that 3 minutes. So I'm going to say my proteins are negative. So now what that leaves us is a little area that you are going to go through and look at a microscopic view. Your sheet actually says copy. So on Google Classroom, you'll see Mr. Rollins, normal slide. I have a slide there of a microscopic view. You're going to kind of copy and paste it. It's underneath the microscope. You'll see the little pointer there. Copy and paste it and put it into this area. What that's showing you is it is normal. There might be a few things, a few cells here and there, but nothing that's causing any concern with this particular patient. Give a qualitative data of that particular slide. So do you see crystals? Do you see shapes? Do you see cells? Do you see anything else underneath that particular one? You don't have to put, oh, I see the arrow of the microscope. I don't need that. Mr. Rollins normal, so he shouldn't have crystals. He shouldn't have shapes or cells or anything else in that. That's there so that when you get to your other patient that you're going to use to compare with, that's where you guys are going to really come in with that qualitative. Do you see any cells? Red blood cells, white blood cells. So as I go through and sketch it, I'm going to say no crystals, shapes, or cells. No cells is what we have. So that will be Mr. Rollins. And like I said, I'll kind of give you guys a photo as well as this video of what that normal test results look like. So from there, you guys are actually going to get a patient profile. So that's going to be kind of day 2. So we're going to save that video for another time. But right now, you guys should have Mr. Rollins filled out. You guys should be able to go through that lab. The beginning of this lab goes through a lot of information for you guys. Not only tells you the function of the kidneys, healthy as well as unhealthy kidneys, but it tells you about all your test results and some possible disorders that could come from if they do have glucose, or if they do have sugar, if they do have chlorides, or if the pH is higher or lower, what could be going on. Because your role with this is once you get your patient, you guys are basically that nurse practitioner, that doctor, and you're going to say, okay, well, here's your test results. Let's research it. What's going on with this patient? What disorder could they have? How can we fix it? How can we solve it? What's my long-term effects? What's my short-term effects? And there'll be a little write-up that you guys will have that based on you being the doctor for that. But like I said, Mr. Rollins is only your control patient. You guys are all going to get different patients along the way. So that video will come a little bit later.